biology final project, and it's about important biological macromolecules. In this video, we will cover carbohydrates, lipids, nucleic acids, and proteins. So to start off, we're going to look at the legend. So, the white wool represents hydrogen, the gray wool represents oxygen, the black wool represents carbon, the red wool represents HO positive, which is hydroxide, and the orange wool represents OH negative, oxyhydride. The presentation will include more elements than these, but for now, these are the main ones. Okay, you llama. Okay, so to the carbohydrate area. Let's get a good view. Here we have some of the prime examples of carbohydrates. The monosaccharides, glucose, galactose, and fructose. I only built representations of monosaccharides because, in all honesty, I couldn't figure out how to properly make a model of polysaccharides and disaccharides. Starting off with glucose, the one on the far left, we have your carbon, your HO positive, the white is hydrogen, and the orange is the OH negative. So, starting off with glucose, glucose is a very important source of energy. During cellular respiration, energy is released from glucose and is used to help make ATP. So, we're going to look at some plants here. Plants synthesize glucose using carbon dioxide and water. And then the glucose in turn is used for energy. And I think everyone knows what that's called. Photosynthesis. And then the glucose in turn is used for energy requirements for the plant. Excess glucose is stored as starch, metabolized by the needle in them. So now we're going to talk about galactose, which is this one right here. You got your OH negative, your hydrogen, at the top. Pretty sure that's gray. So that's the oxygen, hydrogen. And then your fructose, which looks a little different. Your oxygen and hydrogen. Your oxygen's over here. OH negative. Yeah. So, galactose and fructose are sugars that are found in two different sources. Galactose from milk and fructose from fruits. All three of these forms of carbohydrates have the same chemical formula, C6H12O6, but they differ structurally and chemically. These are called isomers. Isomers are when you have the same chemical makeup, but different structures. Different structures and arrangements of functional groups around the asymmetric carbon. All of these monosaccharides have more than one asymmetric carbon. So disaccharides, di meaning two, form when two of these monosaccharides undergo dehydration reaction. During this, the hydroxyl group of one of the monosaccharides combines with the hydrogen of another monosaccharide, releasing a molecule of water and forming a covalent bond. One common disaccharide is lactose, which consists of the monomers glucose, far left, and galactose, middle. It is found naturally in milk. Another one is sucrose, or commonly known as table sugar, which is composed of glucose and fructose. Now let's talk about polysaccharides. Poly meaning many in a long chain of many monosaccharides linked by covalent bonds, and it can contain different types of monosaccharides. Some examples are starch, glycogen, and cellulose, and I hate saying this word because I can never pronounce it, but chitin, C-H-I-T-I-N. Plants can synthesize glucose, and excess glucose is stored as starch in many parts of the plant, including seeds and roots. When anything eaten is brought back to glucose. Cellulose is another one of the most abundant natural biopolymers of the cell, which make up the cell walls of plants, and they're mostly made up of cellulose, which provides structural support. Cellulose is made up of glucose monomers that are linked by particular carbon atoms in the glucose molecule. Lastly, let's look at some spiders here. Carbohydrates serve as different functions for different animals. Arthropods have an outer skeleton called an exoskeleton, which protects their internal body parts. The outer skeleton is made up of the macromolecule chitin, which is a nitrogenous carbohydrate, and it is made of repeating units of modified sugar containing nitrogen. Now let's go on to lipids. So, with the lipids, we're going to be introducing CH3, methyl, and CH2, hydrogen monohydride, which I will just be referring to as CH3 and CH2. So when you hear lipids, you think of fats and oils. This might bring a negative thought due to our culture of always demonizing fats and oils, but our bodies actually require fat in order to maintain life. Just some general facts about lipids. They are largely nonpolar. Nonpolar molecules are hydrophobic or insoluble in water. Lipids perform many functions within cells, like store energy for long-term use in the form of fats. They also provide insulation from the environment for plants and animals. They help keep aquatic animals and birds dry by forming a protective layer over the fur or feathers because of the lipids being hydrophobic, which creates a layer keeping them dry from water, like ducks and otters and stuff like that. Fatty acids can be saturated or unsaturated. First, we're going to look at the saturated fatty acid, which is this one right here. So, pretty much I'll start out with HO positive, and then you got your carbon chain with your CH2, and your oxygen right there, and it ends with your CH3. Notice, before we dive too far into it, notice how saturated and unsaturated fats look almost identical up to this point. Now this one ends with CH3. This one branches out with 2 hydrogens, and then your CH2 plus carbon up to CH3 carbon. So, back to saturated fats. They solid temperature. For example, when you let the pan cool down before you wash it, that the fat has solidified. Saturated fats are obviously found in animals, but they're also found in plants. Store fat or oils in seeds and is used as a source of energy for embryonic development. Let's move on to unsaturated fatty acids. Unsaturated fats are liquid at room temperature and are called oils. If there is one double bond in the molecule, then it is known as a monosaccharide fat, for example, olive oil. If there are more than one double bond, then it's a polyunsaturated fat, for example, canola oil. So what that is referring to is this little hydrogen chain piece right here is what keeps it liquid at room temperature, and that is why saturated fats are not liquid at room temperature. They're in fact solid, and it is all due to this little chain right here, to my knowledge. Unsaturated fats or oils are usually made up from plants and contain unsaturated fatty acids. The double bond causes a bend that prevents fatty acids from packing tightly, right there, and keeping them liquid at room temperature. Unsaturated fats help to improve blood cholesterol levels, where saturated fats contribute to plaque formation in the arteries and increase risk of heart attacks. Now let's look at phospholipids, which is this big guy right here. Let's get that into Phospholipids are the major constituent of the plasma membrane. Like fats, they are composed of fatty acid chains attached to a glycerol or similar backbone. Instead of 3 fatty acids attached, there are only 2 fatty acids, and the third carbon of the glycerol backbone is bound to a phosphate group. A phospholipid has both hydrophobic and hydrophilic regions. Acid chains are hydrophobic, right there at the bottom, include themselves from water, whereas the phosphate, right there, is hydrophilic and interacts with water. They're surrounded by a membrane that has a bilayer of phospholipids. The fatty acids of them face inside. So the fatty acids are inside, away from the water, whereas the phosphate group, up here on the outside, can either face outside or inside the cells, which are both aqueous. So waxes, let's move over here, are made up of a hydrocarbon chain with an alcohol negative OH group and a fat waxes lanolin. Plants are just the coating on their leaves that helps prevent them from drying out. So this beehive would be susceptible to drying out and or collapsing if it didn't have the fats from the lipids there. And plus this little bee right here, he would get wet and pretty much die if it wasn't for the phospholipid layer on its wings and all that. So I think I covered enough of the lipids. Let's go on to nucleic acids. So just some general nucleic acid facts. They are a key macromolecule in the continuity of life. They carry the genetic blueprint of a cell and carry instructions for the functioning of the cell. The 2 main types of nucleic acids are DNA and RNA. DNA is the genetic material found in every living thing. The other type is RNA, which is mostly involved in protein synthesis. The DNA molecules never leave the nucleus, but instead use RNA as an intermediary to communicate with the rest of the cell. DNA and RNA are made up of a monomer, nucleotides, which you see here. Nucleotides, they have their and phosphate with your over here. So they're, like I said, they're made up of a nitrogenous base, pentose sugar, and a phosphate group. Each nitrogenous base in a nucleotide is attached to a sugar molecule, which is then attached to a phosphate group. The nucleotides link together by phosphodiester bonds to form a polynucleotide. So let's go look at some DNA. This is the best I can make a double helix in Minecraft without making it ultra massive. So I did my best with the colors, as I will describe what each of them mean in my notes, but each color represents a different part of the DNA, and they get linked together to make the genetic code for making stuff. So, DNA has a double helix structure. It is comprised of polymer nucleotides that are strands. The strands are formed with covalent bonds between phosphate and sugar groups of adjacent nucleotides. The alternating sugar and phosphate groups lie on the outside of each strand, forming the backbone. The nitrogenous bases are stacked in the interior like rungs of a ladder, or I should say rungs of a twisted ladder, because essentially you can climb up and down, but it'd be hard because it's twisted. So These bases pair and they are bound to each other hydrogen bonds. To each other with hydrogen bonds. The bases pair in such a way that the backbones is the same the whole way up. The rungs on RNA, so let's look at some RNA, consists of alternating patterns of cytosine, which is the blue one, guanine, green, adenine, yellow, and uracil, orange. The rungs on DNA are the same, except instead of uracil, it's thymine. Some of the traits of DNA is when it carries genetic information, it remains in the nucleus. It has the double helix structure, its sugar is deoxyribose, and its, hate this word, pyrimidines are cytosine and its purines are adenine and guanine. For the RNA, its function is involved in protein synthesis and it leaves the nucleus. It usually is a single-strand structure. Its sugar is ribose, and its pyrimidines is cytosine and uracil, and its purines is adenine and guanine. Information flow in an organism is from DNA to RNA protein. DNA dictates the structure of mRNA in a process known as transcription, and RNA dictates the structure of a protein in a process known as translation. This is known as the central dogma of life, which any organism, however, does not apply in connection with viral infections. Now let's go to our final macromolecule, proteins. So before I start reading my notes, these four structures are amino acid. Notice how they have there's 20 amino acids, but they all have similar structure, as you can see by these four that I made. So the quartz brick is going to represent the carboxyl group, and then the light gray wool instead of CH2 is going to mean the amino group. So you got your CH3 amino group, carboxyl group, hydrogen, and then carbon. More CH3, CH, just regular CH. Then your carbon, hydrogen, carboxyl, amino. This one has amino, CH24, C, amino group, carboxyl group, hydrogen. Lastly, carboxyl CH2, C, carboxyl group, amino group, hydrogen. So I'm going to get in the view of all four of these so that you can look at it while I read my notes. So proteins are the most abundant macromolecules in organic organisms and have the most diverse range of all the macromolecules. Proteins have several structural, regulatory, contractile, protective functions. They also may serve in transport, storage, or they may be toxins or enzymes. Which cell could have one cell have 1,000 different proteins? They all have unique functions. They all have different structures and functions. They are, however, polymers of amino acids arranged in a linear sequence. Proteins have different shapes and weights. Some are globs in shape, while others are fibrous in shape. Hemoglobins are an example of the glob shape, while collagen found in our skin is fibrous. The shape of the protein is critical to its function. They are subject to permanent changes with change in temperature, pH, and exposure. All proteins are made up of different arrangements and of the same 20 kinds of amino acids. So depending on how those 20 amino acids are arranged and the structure of them will dictate what the protein function and structure is. Amino acids are the monomers in protein. Each one has the same fundamental structure as seen by the replicas, and it consists of a central carbon bonded to an amino group and a carboxyl group, which I showed you earlier, and a hydrogen atom. The sequence and number of amino acids determine the shape, size, and function. Each amino acid is attached to another with a covalent bond, known as a peptide bond. The products formed by such a link are called polypeptides, which is a polymer of amino acid. The term protein is used for a polypeptide or polypeptides that have been combined together and have a distinct shape and function. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. The primary protein structure is a sequence of a chain of amino acids. The unique sequence and number of amino acids in a polypeptide chain is its primary structure. The unique sequence for every protein is ultimately determined by the gene that encodes the protein. The folding patterns resulting from the interaction between the non-R group portion of amino acids give rise to the secondary structure of the protein. The most common are the alpha helix and the beta pleated sheets. So this is the alpha helix, nothing special, just a helix, and the beta pleated sheets, which is like a folding sheet of paper. The pleated sheets are formed by hydrogen bonding between the atoms on the backbone of the polypeptide. The R groups are attached to the carbon. The pleated segments align parallel to each other, and hydrogen bonds between the same pairs of atoms on each of the aligned amino acids. The helix and sheet structure are found in many globular and fibrous proteins. So according to the textbook, the unique three-dimensional structure of the polypeptide is known as a tertiary structure. This is caused by chemical interactions between various amino acids and regions of the polypeptide. Primarily, the interactions among R groups create the complex three-dimensional structure of a protein. They may be ionic bonds formed between R groups on different amino acids, or hydrogen bonding beyond that in the secondary structure. When proteins folding takes place, the hydrophobic R groups of nonpolar amino acids lay in the interior of the protein, whereas the hydrophilic R groups lay on the outside. So, the gray represents your alpha helix, and the white represents the pleated sheets. And as said in the notes, inside, outside. One is outside, one is inside. These former types of interactions are also known as hydrophobic interactions. Also, according to the textbook, some proteins are formed of these subunits forms the quaternary structure. Between subunits help to stabilize the overall structure. For example, hemoglobin is a combination of 4 polypeptide subunits. To recap, the primary structure is a sequence of a chain of amino acids. So, a sequence chain amino acids. The secondary structure is hydrogen bonding of the peptide backbone causes the amino acid to fold into a repeating pattern. The tertiary protein structure is a 3D folding pattern of protein due to side chain interactions. And then quaternary protein structure is protein consisting of 1 or more amino acid chain. So let's go to the fun part and talk about protein function. So we're going to look at digestive enzymes first. The lava is going to be the digestive enzymes, which can be made up of amylase, lipase, pepsin, and trypsin. So, and what that does is it helps digestion of food and catabolizes the nutrients into monometric units. So let's put some food in here. So, as you can see, it gets spit out into the enzymes, which get digested. Another form of protein, another function of protein is transport, done by the hemoglobin and the albumin. I'm probably butchering that saying, but so it carries substance in the blood or lymph throughout the body. So, for example, it could take the subunits or the monometric units from the enzymes and then transport them to other places. Third, we're going to look at defense. Let's just make sure that we're on easy. So, another job of protein, depending on its function or its structure, is defense. And it protects the body from foreign pathogens. So here we have some iron golems to represent the immunoglobulins. Sorry about my pronunciation, but so here they are. They're just chilling until a foreign pathogen comes in, represented by the pillagers. As we can see, the proteins are defending the body from the foreign pathogens. They do a pretty good job of it, if I do say so myself. Let's just do some more for good measure, just to really demonstrate it. See how much they can handle. So, they're really taking some damage in there. So as you can see, the foreign pathogen has one, which sometimes can happen. Like with an advanced virus like COVID-19, the proteins cannot defend so easily. For one, it could be too many of the foreign pathogen in the body, or it could just be because it didn't know how to fight it. In this case, it did know how to fight it, but there was just too much of it. So, our patient has died of the foreign pathogen because it could not defend. Anyways, let's go over here. So, I didn't really know how to really set up the hormone function of protein, but another function of protein is hormones, which coordinates the activity of the different body systems. This could include like growth spurts, like what starts puberty. It's a chemically signaling molecule, very small proteins or steroids secreted by the endocrine cells. And they help include growth, development, metabolism, reproduction. And then like insulin is a protein hormone that helps regulate blood glucose. Some people can't do that, and it's called having diabetes, when they're can't produce the right amounts of insulin. So let's say our representation of the body right here has too much sugar in it. Our person has insulin control problems, and the sugar is just running rampant. So what should happen is their body should secrete some insulin, which would have the sugar get under control. So we're going to say that this is what shoots out the hormone, the dispenser, and we're going to try to control the blood sugar with the insulin. So the body's just doing its job, using the insulin to take control of the blood sugar in the body. So, since it's a hormone, I used like potion stuff to act as it. So, your little proteins that have the hormone on it, for example insulin, taking control of the glucose levels. I could have did a better job explaining that, but that's the best I could come up with. Proteins also have many other functions like storage using legumes, storage proteins, and egg whites. And it provides nourishment and early development of the embryo. Contraction with myosin and actin. And digestive enzymes. All proteins are the most diverse of all the macros. It's pretty crazy. I didn't learn till after I learned it in class. I didn't know that proteins had so many functions and shapes. So, and then another thing to think about too that I didn't really take into consideration is that say you go to the gym all the time and you're really into supplements. You get that BCAA drink to help replenish muscles. I did not know that amino acids were basically the building blocks of protein, and that's why it helps reduce muscle soreness. So that BCAA drink that you're drinking is essentially helping replace all the shredded proteins in your arms and legs and wherever else you're working out. So that is my presentation. Just gonna do a little overview again. So you got your carbohydrates, your three main monosaccharides that pretty much supply energy for ATP and energy for plants, and it actually makes up the exoskeleton of arthropods. For lipids, they store energy. Saturated fats clog the arteries, while unsaturated fats help maintain cholesterol in the blood. Phospholipid bilayers help just regulate the water stuff of the cell. Sorry, I blanked on that. All in my notes in front of me, but main thing is, or something I'll definitely take with me after the class, is that the reason why beehives are protected and otters and birds and other aquatic animals is because of the waxes, which are made up of lipids. Did not know that's why ducks could get in and out of the water. I was always wondering why they didn't really, they got in the water but they didn't really get wet, they would never be dripping. It's because of that lipid bilayer on their fur. And then I pretty much did know a good bit about nucleic acids, RNA's protein synthesis, while DNA is in the nucleus and carries the genetic code in this protein. So, in proteins, amino acids make up proteins, four primary, secondary, tertiary, and quaternary structure. But I did not know that proteins were enzymes, toxins. They are used in transport, defense, and hormone regulation. Had no idea that what was fighting all the bad stuff in our body was hemoglobin or immunoglobulins made up of proteins. Biology is pretty fascinating. So that's my presentation. Hope everyone has a good one.